This is Russ with Thermo Scientific, here to talk to you about our genome editing solutions for cell line development and engineering. Now, as a genome editing service provider, we have a ton of experience. We can do any edit in any cell line. We work with CRISPR-Cas9 as well as TALEN nucleases. We can do knockouts, small knock-ins like a SNP correction, large knock-ins like an insertion of a gene, a reporter, or other large donor. We use award-winning tools to get this done, like our Cas9 version 2 and our lentiarray CRISPR knockout libraries. And then at the end of the day, we try and provide a very customized solution because your biology is your own and you know what you're trying to interrogate at the end of the day. And it's not just an edited cell line, it's biology. So it's a very consultative process to make sure that the edits we're doing and the work we're doing is fit for purpose to deliver the biology that's going to move your programs forward. Now, I mentioned, our ability to be consultative. This is critical in what we do, especially when you're going after difficult biology. Making sure that we work together to scope out the needs, to understand the different technologies and options available to you, and any downstream concerns around biology, the model, the endpoint, etc. Every project is unique for this reason. We approach everything with our expertise, but with fresh eyes, with your needs in mind, not our needs, not what we've done, but how can we solve your unique problem. Everything we do for that reason comes with a dedicated project manager. As we become an extension of you, we understand communication is paramount. So that project manager is going to be there for regular updates, regular calls, any communication. And as you progress through this work, everything's milestone based. So you're going to get reports at the end of every milestone, and you will have the decision to move forward or stay, and that will be a technical discussion that we have that you will be the owner of. We can do a lot to help mitigate risk by optimizing cell culture conditions, transfection methods. We can do upfront things like clonality assessment with difficult to grow cell lines. And last but not least, I mentioned this a couple times, but I can't drive it home enough in this space. We validate for your needs. If we can't move your biology forward, we weren't successful. And because if we can make you successful, we're successful. Taking a look at the workflow for gene editing, it starts with the design and the build of the guide RNAs, the oligos for donor templates, using our TrueCut Cas9, and then delivery methods. We can work through that with you or do some of that work internally to understand the ideal transfection conditions or transduction conditions. And then once we've done that, the real work starts, where we need to now isolate and screen and expand your cells of interest. And so we do single sorting, clonal screening, and expansion of clones with extensive characterization and validation each step of the way. We are confirming all your sequences with NGS. We have a variety of off-target systems to make sure that we've edited the loci you want and not other loci. And then again, the biology, making an edit without functional biology is irrelevant. So we work to make sure these cells are functionally characterized to your endpoints. And then if you're working with more difficult cell lines, we have a lot of characterization tools for stem cells and other cell lines as well. I mentioned our ability to mitigate risk with clonality assessment. So we can do that upstream to help get you a clonal population. We can do limited dilution cloning if your cells are challenging to work with, or FACS analysis. And we've optimized some different workflows to improve our ability to do FACS on difficult to work cell lines like iPSCs. We can now confidently, routinely sort these as single cells per well, and it's drastically improved our ability to get difficult edits in difficult cell lines. We screen everything through NGS. And in addition to NGS, on-target and off-target, we have a cool technology called TegSeq. And what TegSeq is an unbiased whole genome analysis for off-target. It's great for looking at rare events. I'd be happy to talk to you more about this technology in more detail. We also can look at genomic instability, gross chromosomal changes. This is, again, more important with particular or finicky cell lines like pluripotent stem cells. In summary, let our team do the heavy lifting. Let us become an extension of your lab, your scientists, and we're here to let you design the solution that we will build for you. We will implement this with the most cutting edge tools. and adapt those to whoever you need to be assured that your biology is what we're going to deliver in the end. And again, we're unique as a service provider in that we're pulling the same kits and tools off the shelf that you are. So there's a very low transfer barrier. We share everything we can. We become an extension of you. We're a tool provider. If our services can make you successful, you'll use more of our tools, and your success is our success. So I encourage you to reach out to your Thermo Fisher partners today, either your bioscience account manager or your cell biology technical specialist, to talk about how our custom biology services can solve your difficult biology. We can do any edit in any cell line, and we'd love to have a conversation about how we can enable your programs to move forward. Thanks again. I'm Russ with Thermo Fisher Scientific.